So just a bit about David. He is an associate professor at the University of Sydney in the Sydney School of Veterinary Science, Faculty of Science. He does a lot around the uni. He teaches wildlife and exotic pet medicine to DVM students and also animal and veterinary bioscience students. He also supervises an array of research students, so from wildlife masters, honours, PhD students, postdocs, all that sort of stuff. He also is my PhD supervisor, so yeah, and he has a lot of interest in his research. So they're quite varied. They include infectious diseases, toxicological analysis, nutritional diseases of wildlife and exotic animals, in addition to ecology and conservation. So yeah, like I said at the end, don't forget to keep your questions. I will have some time, and yeah, use those two type of methods if you can't, you know, kind of video yourself or ask questions. But I'll let David take it from here, and he'll be able to show you his presentation. So.
Well, thank you for inviting me tonight, and it'd be good to talk to you and hear what your questions are. The title of my talk is Now That I Found It, What Does It Mean? Pathogen Hunting in the Modern Age. But I'm going to start by just talking to you about my history and how I got to where I am, so that veterinary students and PhD students that are interested in wildlife diseases can see at least one rather circuitous route to achieving their goals. So I always encourage people to start out early and to really get passionate about what they're interested in. And my passion started when I was in 9th grade and was able to go on an owl trip with the Delmarva Ornithological Society in the middle of the winter, where we found 7 species of owls in one day. And finding the little saw-whet owl in the tree just where it was supposed to be was probably one of the keys to what I've done for the rest of my life. I entered the University of Chicago in a long time ago and obtained a degree in biology. But along the way, I thought I might go for combined Bachelor of Arts and Masters of Science in biochemistry. But I was still very passionate about going to vet school, and I decided that wasn't the best for me. I did an honors project, which didn't turn out completely very well, and I decided I was never going to do research again. But during that time, I did work in a research facility with animals, laboratory animals, and spent a considerable amount of time volunteering at a veterinary hospital in order to get the experience that was necessary at that time to get into vet school. I went to vet school at Cornell University, which is in Ithaca, New York. It's along the far south end of one of the Finger Lakes, Cayuga Lake, up on a hill. Absolutely beautiful place to be, except when it's about -30 and the wind's blowing hard out of the northwest, like it sometimes does in the middle of the winter. I was very interested in pathology very early on. And then I spent a summer doing research on the impacts of oil ingestion on seabirds at Mount Desert Island in Maine. And that really reignited my interest in research. And it confirmed that my long-term objective was to work with birds and with wildlife. We didn't know an awful lot about wildlife medicine at that point, or even about wildlife diseases. And so everything we were doing really at that point was pretty cutting edge, even though it was the same sort of things that people were doing in other species. No one had ever applied it to them. And so everything that we saw was new, and there was an awful lot to learn. And we stumbled along, but we made progress and headway. Wanting to do birds and wanting to do wildlife, what's the first thing I did? Was I started out mixed animal practitioner in southern Pennsylvania. I guess I thought I was going to be James Herriot or something and do all creatures great and small. And so I was seeing small animal practice in the afternoons, large animal during the mornings, large animal at night. Found out that being a large mixed animal practice in southern Pennsylvania meant working typically 6 to 7 days a week, being on call 3 to 4 nights a week, and basically having no other life than doing that. It was a beautiful place to work, but it was very, very hard and difficult. And it became clear to me that it's going to be very difficult to become a master of all species, especially in that kind of a setting. And so I moved to small animal practice and exotic animal practice in Rochester, New York, where I worked at the Henrietta Animal Hospital and the Pittsford Animal Hospital. One of the things, though, that impressed me at the time that I was a mixed animal practitioner and has stayed with me ever since then was that during that summer that I worked there was an emergence of rabies, and the raccoon rabies was taking off and spreading up the East Coast. And there were over 70 animals in the county that I worked in alone over the first 6 months of the year that had rabies. And so virtually every single animal that you interacted with, the first thing you did was assess whether it had neurologic signs because of the risk to yourself. But in these days of COVID, it helps to remember that emerging diseases have been around for a long time, have very, very significant impacts, and will continue to be a very important part of what we do as wildlife veterinarians into the future. I decided after 6 years of being in private practice that I wanted to go back and do a PhD. And the reason for doing that was that, again, a novel infectious disease, avian polyhomovirus, recently emerged in captive bred budgerigars and also in a number of other species of birds. And the thing that perplexed me and I wanted to find out and understand was why these pet shops, they'd buy these expensive conures and macaws and bring them into the pet shop, feed them up, they'd look great for 2 weeks, and then the next day they'd come in and find them very sick or dead. And why was that happening? And that was something that drove me to return to get my PhD at Texas A&M University in Texas, and to work in the Shubot Exotic Bird Health Center, which was a center that was founded or endowed by an aviculturalist who was quite wealthy, who was interested in trying to understand the diseases that were impacting his collection. So my PhD was on avian polyhomovirus, and I graduated in 1992. During that process of PhD training, it became very obvious to me that getting a PhD was an entirely different experience than getting a veterinary degree, and it resulted in a whole nother level of skill sets beyond what you got as a veterinarian. In this day and age, we do some research in the DVM curriculum, and so you get more exposure than what I would have had. But nevertheless, the type of training that you get as a PhD in terms of learning on your own, critically assessing the data that you see, critically assessing all the data that other people have generated, coming up with your own ideas as far as what kind of research should be done, testing them out, and overcoming all the challenges that come with that. Creates a very, very different set of skill sets, and also puts another perspective on life that's quite different than being in veterinary medicine. I found, and I still believe more than anything else, that interdisciplinary research, it brings the greatest rewards. And I've always reached across, as a clinician, I've always worked with pathologists and virologists and epidemiologists and toxicologists. And when you've got a challenge and you've got a problem, it's a lot easier to reach out to someone that's already an expert in that field and to learn from them than it is to reinvent the wheel and because it just takes so much more time to get up to speed. and having a great team, whether you're working in one area in the lab or whether you're working across disciplines in the field. So if we're working with infectious diseases in the field, I'm going to want to have an ecologist in there. I'm going to probably, want to have a biologist and maybe even somebody that's an expert in mapping, and maybe even somebody that is an expert in soil or rocks or plants. All these people come together to generate great outcomes. And if you try to work independently or in your own silo, then you're not going to be successful, especially in this day of an age of really competitive funding that's out there. And the bottom line is nobody's going to hand you anything unless you work hard for it. And so if you work hard, if you have an inquisitive and questioning mind, and think about what you're doing, then you will have good luck. Good luck is not an accident. It's an outcome of working hard and thinking, and always thinking about what you're doing. So after that, I was very fortunate to be able to get a position at Texas A&M University, which had a very active zoological medicine service where we saw both client-owned animals and wildlife. And I finally sort of became that doctor of all creatures, great and small. And we saw ferrets, rabbits, and rodents, all kinds of exotic birds and reptiles. But we had both a large animal and small animal component to our service. And we saw lots and lots of South American camelids, llamas, and alpacas. We had, everybody's watching this program, I guess, on TV. Well, I've lived through it. I've seen many, many crazy people with their big cats come into our hospital. We've lived through the great rat tide adventure of the early 90s, which was when everybody thought they were going to get rich raising ostriches and emus and rheas. And I saw all kinds of unexpected and weird diseases in them, and saw some amazing wildlife, and some amazing wildlife that was kept as pets. And this was down the lower left-hand corner, that's a coatimundi. And if you ever get a chance to even see those guys, they're just amazing in the wild, but are incredibly amazing when they're humanized. Doing research is really a privilege, and there's not many jobs where you can do as much research as what you'd like to do. And in this case, when I was at Texas A&M, I had a lot of clinical responsibility, had a lot of didactic teaching responsibility, and research really was what you did on your spare time sometimes. And that means that there's no such thing in academia as a 40-hour week. And so if you intend to go that route, you will be spending more time at work than the average tradie who comes home at the end of the day. From there, I was fortunate enough to be selected to come as the director of the Avian, Reptile, and Exotic Pet Hospital, 2006, and I've been at the University of Sydney since then. I'm no longer the director of the hospital. I don't do as much clinical work as I used to, but I still do a fair amount of teaching. I think one of my favorite teaching experiences was the Master of Wildlife Health and Population Management, where we got to combine veterinarians as well as wildlife biologists and to work together to show all the different aspects of the kind of tools that you need in order to investigate wildlife decline and to measure wildlife population, the health of wildlife populations, the conservation tools, as well as the veterinary tools. And that's been very exciting to do, and it's been great to see that the students that have graduated from there have gone on to do a lot of wonderful things. And I've been very fortunate to do some amazing research with a large number of species, ranging from amphibians to koalas and birds and everything in between. So I've been blessed in that regard. So if you want to become a wildlife veterinarian, these would be my suggestions to you. First of all, you need to demonstrate a passion in this field as soon as you possibly can. And what does that mean? It means that nobody's just going to hand something over to you. You need to go out and find, if it's a job and it pays you, that's great. But oftentimes it means that you volunteer somewhere. So for me, it was volunteering at an animal hospital that was an hour and a half train ride from where I was when I was an undergraduate. It meant working in a laboratory animal care facility where I worked with primates. It meant many instances, and also going up to the zoo and helping with postmortems that were being done there. All, again, pretty much on my time, so that I got the experience, so that when I applied to vet school, or when I started talking to people about what I wanted to do, I could actually show that I'd already made efforts to reach those goals. I think the other thing to remember is that the veterinary degrees give you incredible flexibility. And as you can see from my career, I mean, I've been a mixed animal practitioner. I've been a mixed small animal exotic practitioner. I've worked with exclusively exotic practices and exotic and wildlife practices. And once you get into those areas, veterinarians can be in major demand because they have to be present oftentimes in a lot of procedures that occur in the wild. And I've had the chance to do research all over the world and be engaged with a lot of really fantastic groups. A veterinary degree with a PhD gives you even more flexibility. And if you don't have a PhD but get a specialty, then again, that can give you a lot of flexibility, a lot of different opportunities, a lot of different doors can open up. But as I tell my students, it's not likely that you're going to go from A to B and graduate from vet school and all of a sudden be a wildlife veterinarian. You usually have to go from one step to the next. And sometimes those steps are sideways instead of forward. So a really good example was one of my students who was bound and determined he was going to become a zoo veterinarian, and he was going to do a residency with Dr. Carpenter at Kansas State. Well, Jim Carpenter doesn't ever take anybody that's just out of vet school. You've either got to have done another residency somewhere else, or you've got to have established yourself in some other way. And Rob, he got to be quite a knowledgeable person about chameleons, and he published widely. He presented at meetings, and he went out and worked in private practice until he could convince Jim Carpenter that he should take him into his residency. And then he did his residency, and at that point, he was fortunate enough to get a job at the San Antonio Zoo. If you want to become a wildlife researcher with a PhD, again, first thing that you need to do is demonstrate your passion, and you can do that with all kind of volunteering stuff. But the bottom line is, if I'm looking for an honors student or looking for someone that's going to be a PhD, the first thing I look at is their WAM. And so you really need to get good marks in school. You need to excel at the classes that you're taking. Get an honors project where you've got lots of hands-on experience. Volunteer if you need to get more. And then once you're in that position, hopefully you'll get a publication out of your honors, and then find the right PhD program for you. And the right PhD program is going to involve talking to lots of people, getting to know them, talking to PhD students that are already in that program or graduated from it. And then when you get there, you just have to work your butt off. And if you do, and you publish, and you apply for grants, and you get money, and you present as many times as you possibly can, then the opportunities will open up for you on graduation. So that's phase one. Phase two of the program tonight is talking about molecular diagnostics and metagenomics, and how these are going to be, how they are, and how they will be really the bones of our diagnostic capability in the future. And they're just absolutely amazing with what they can do. And the first one I want to talk to you about is pseudocyd adenovirus infection in the critically endangered orange-bellied parrot. This was done by one of my wildlife masters. This was published by one of my wildlife master's students, Nian Yang. And it brings up a theme that I want to talk about throughout the rest of this presentation, and that is metagenomics and molecular diagnostics are very good at identifying pathogens, identifying new pathogens, identifying pathogens that you might not expect to be there. But the next step always with these techniques is figuring out what that means, because there are so many different organisms that are crawling around or running around or hanging around in our bodies and all other species that don't cause any disease, that just necessarily finding one, even if it's known to be a pathogen under some circumstances, doesn't necessarily mean that it's important in the disease of that animal or that's having an impact on that animal in general or the populations of that animal. So I'll give you a little bit of background about orange-bellied parrots. They're one of two migratory species that we have in Australia. They're a specialized parrot that only is found along the coastal areas of the southwest corner of Tasmania, where they breed, and the coastal areas of southeastern Australia. And they used to extend from South Australia, their wintering habitat from South Australia all the way up to Sydney, where they're found predominantly in saltwater marshes or on the edges of saltwater marshes. There used to be tens of thousands of them, and now there's only a couple of dozen left in the wild. And to keep them from going extinct, they've established breeding colonies, and they have 5 captive breeding centers, sites. And these birds maintain the genetic diversity that's present in the population at this point, and the animals that they produce that they don't need for captive breeding are released into the wild to supplement the wild population in the hopes that we can get it back to a point where it can be self-sustaining. In 2016, in the summer, at the end of the summer, there was a mortality event at the Adelaide Zoo. And 6 birds died there during a severe heat wave. During this time of the year, most orange-bellied parrots, all orange-bellied parrots, wild ones, are down in the southwest corner of Tasmania where it's damp and it's cool. Adelaide is an entirely different environment. It gets very, very hot right there. And any bird that's in a nest box at the Adelaide Zoo at the time was getting very, very hot and overheated. And most of these birds died with an infection of their lungs caused by a fungus called Aspergillus. But some also had these purplish inclusions that you can see with the arrows in the nuclei of the tubular cells of the kidney. And these are very, very characteristic of adenovirus inclusions. And so it immediately became an issue as to what was causing this, and is this a threat to the captive population of these birds? So I was sent some tissues, which extracted DNA, and then we used what we call degenerative primers with PCR to amplify sequences of adenovirus from those tissues. And what degenerative primers are, PCR uses 2 sets of primers, one that binds to one strand of the DNA and one that binds to another strand of the DNA at a certain distance, a certain number of nucleotides separate those primers. And then they use a Taq polymerase, which grabs onto the primer and duplicates that DNA of the exact size between those primers to a point where if you run it out on a gel, which you can stain with different things, this is lithium bromide gel, and you can see the bands which are positive samples. Degenerative primers are primers that are designed to pick up all known viruses or bacteria that you're after. And then what they do is people have sequenced certain genes, in this case the DNA polymerase, And not all the DNA polymerase sequences are identical, but they're very, very close to each other. So degenerate primers are actually a mix of several different primers that are almost identical to each other, but have several mutations in them so that they'll match up with whatever adenovirus is in there, no matter which adenovirus that you're looking for. And we, in fact, did find adenovirus in these tissues, and we sequenced it, and we went to GeneBank, which some of you are surely quite familiar with, which is a repository of all known DNA sequences that have been put into GeneBank anyway. And there's a program called BLAST that you can compare your sequence to all the sequences in GeneBank. And what we found was pretty much 100% hit to pseudocyd adenovirus 2. And pseudocyd adenovirus 2 has never been found in Australia before, so that was a discovery of concern. It had been found in US and Europe in parrots. All of these parrots, of course, have been imported into US and Europe because there's no native ones there. And so presumably it came from somewhere else. Did it come from Australia? We don't know, but it might have. What we do know is that it's not particularly pathogenic, at least in descriptions of US and Europe. And we saw that in this case, that it was causing disease, but it was causing disease in some already very sick animals. So what did this mean for the orange-bellied parrot? Well, if it turned out that the orange-bellied parrots at Adelaide Zoo were the only ones infected with this, then they would be quarantined and their birds would either have to be tested and shown to be negative, which we didn't know how to test for it then, or they would have to be taken out of the breeding program. So that was quite a concern. So what we did next was to screen other breeding colonies of the orange-bellied parrot for adenovirus infections. And what you can see in this table just down below is that every one of the breeding populations that we tested was positive for pseudocyd adenovirus 2. And so it was widespread, and we were able to get a hold of some birds that had been released to the wild, but then were brought back into care for the winter. And these birds were also positive for adenovirus. So it meant the wild birds had also been exposed. So it was enzootic in these different collections. If you move one bird from one place to another, it'd be just going from one aviary where the virus was to another aviary where the virus was. And so therefore it was no longer considered to be a significant pathogen and one that was going to impact the movement of birds around in the breeding colonies and impact the release of the birds that were being released into the wild. And in the end, we pretty much decided that the only time that this adenovirus was going to be a problem was secondarily when birds were experiencing other severe stressors, and that healthy birds probably weren't going to be impacted by infection at all. However, we did have some preliminary data that suggested that possibly in the colonies with the highest prevalence of infection, that fertility, hatchability, and overall livability of the offspring might be diminished in these larger colonies, or in these colonies with a higher prevalence of infection. And so that remains to be determined whether it may have some impact on these birds or not. Additional work that's been done after that showed that pseudocyd adenovirus 2 is in fact widespread in aviculture in Australia and in the Burke parrot and the scarlet-chested parrots, as well as other species. It's widespread and very, very high prevalence in these little backyard aviaries. And so it's possible, at least, that this adenovirus got into the orange-bellied parrots because some of them are bred in facilities where there's other species of birds, or it's possible that they could even have be the host of these species. And we found very similar adenoviruses in wild birds now in lorikeets and galahs and other species. And so probably pseudocyd adenovirus is an Australian virus that has spread in the wild in certain species of parrots and we don't know how it got into the orange-bellied parrot, but they may have been one of its natural hosts historically. So the next thing I want to talk about briefly, very briefly, is some work that Jess has done and how we have used next generation sequencing to understand how a particular organism that can't be grown in the lab and can't be studied in traditional methods causes disease, and try to better understand its biology. And so just to get you filled in on the background of this, the organism that Jess is studying is an Enterococcus species that had never been discovered before. It is the cause of fatal infections in captive breeding colonies of 2 critically endangered reptiles on Christmas Island, the Lister's gecko and the blue-tailed skink. And why it's such a concern is that these animals are only present in the captive breeding colony. They are no longer found in the wild. And so an infection in the captive breeding colony would be quite a significant event that could endanger pretty much the most important population of these animals. It's also now been identified in invasive geckos on the island, which appears that they are the ones that are maintaining it in the wild and are the source of it for the captive breeding colony. So this is what, sorry, let's go back here. This is what it does to the lizards. You can see this lizard is infected with it, has a huge bulge on the top of its head. Based on Jess's work, this infection could occur as a result of biting, which does occur between animals during mating and probably during territorial aggression. Or it may be that they get it through other means where it colonizes their oral cavity and then it disseminates locally into the tissues under the skin and around the skull, where it essentially invades into those tissues, dissolves them, and eventually disseminates to other tissues where it causes abscesses. And you can see the little gray abscesses in D here. And these are just massive colonies of this Enterococcus, which we can see in E, that are surrounded by this thick biofilm layer. So what did we want to know? We can't grow it. So what we wanted to know was, first of all, what is it? Is it anything that we've ever seen before? And we amplified ribosomal DNA, which you can do from all bacteria using specific types of primers. And we sequenced that, which gave us an idea that it was an Enterococcus. And then we used specific primers that target specific genes and sequences for the enterococcal proteins, amplified those, and were able to do a molecular phylogeny that showed and proved that this was, in fact, a novel species that was related to other enterococci, but had never been described before. And then because we couldn't grow it, we couldn't determine what kinds of nutrients it needed using traditional bacteriological methods, and because we had no idea which type of antibiotics it was sensitive to, and we had no idea why it formed these this massive biofilm that it did, and how it avoided the immune system. We used next generation sequencing to amplify the DNA from the organism and then identify the bacteria in it. So this is next generation sequencing for dummies, which is about the level that I understand it. When you do next generation sequencing, you amplify all of the DNA that's in your sample. So that includes host DNA plus the pathogen DNA and any other pathogens that might be in there that you didn't even know that were there. Once you've got all your sequences amplified, then you use programs to subtract out the host DNA, or hopefully you have the host DNA sequence available or another host that's very similar to it. And you assemble the overlapping sequences that are left over to form what we call contigs. And then you look to find a contig that's the right size for what you're looking for, and that it contains sequences that are similar to what you expect. And then you can use many different programs to search your new sequences for the genes that you're interested in. And so this is what Jess did. And what she was able to do was to map the majority of the genome, probably the vast majority of the genome. And with this information, we were able to get a better understanding of its genetic history, which includes, which means that we have a better understanding of its phylogenetic relationship to other enterococci. We've been able to identify virulence factors that may be associated with it causing the disease that it does, genes that are associated with biofilm formation. And now we know, based on its genome, probably which antibiotics it's resistant to and which ones it's probably sensitive to. And we've been able to understand a lot more about its metabolism and why it may not be able to grow in cell culture. And then comes the unexpected. And one of Jess's colleagues was playing around with the data and found a novel, one of our colleagues, one of our collaborators, found a novel papillomavirus in the next generation contigs that we had there. And so we found a new virus. We started screening some of the other tissues from the other lizards for this, and we were finding it, and we started sequencing them. And we realized, in fact, that there was more than one virus there. There was 2 papillomaviruses. And we found that they were present both in the Lister's gecko, endangered species, and the 2 invasive geckos on Christmas Island. And importantly, that was also present in these geckos on Cocos Island, which is islands not too far from Christmas Island, where they were thinking of translocating these animals, the Lister's geckos and the blue-tailed skinks at one point or another. So what did this mean? So we found a pathogen. Is it significant? Is it having any impact on these animals? And the answer is probably not. But the first thing that we got quite excited about was not so much its disease impact, but just its biology and what it told us about the evolution of papillomaviruses. And the one thing that was very unusual about this was we found it both in skin samples and also in viscera. And papillomaviruses, prior to a few recent discoveries, have only been found in epithelial surfaces and mucosal surfaces and weren't thought to be able to replicate in other tissues. And we have evidence now that, in fact, it may be able to replicate in tissues such as liver or possibly the biliary epithelium or something of that nature. We also found that it was a unique virus in that its most closely related virus came from a fulmar, which is a northern Atlantic pelagic species of birds, and it wasn't really related to the reptile viruses that have already been found in this group of bird and reptile papillomaviruses. And it's a very, very primitive virus, having the fewest number of genes in this group and some of the fewest number of genes of all the papillomaviruses. So it gives us some idea of what's actually necessary for papillomaviruses to replicate. So that was very interesting. But we don't think it does much in these animals. Some papillomaviruses cause warts, and other papillomaviruses not only cause warts, but they can lead to malignant transformation in a lot of different species, including fish and a number of different species. And particularly in women, this is a major problem, where the human papillomaviruses are important causes of cervical cancer. But no papillomaviruses have ever been seen in these geckos. And given the fact that it was found on Cocos Island and also on Christmas Island, meant that if we were moving our geckos from one island to the other, they'd already been exposed or would likely have possibly been even infected with this virus, and having it on Christmas Island and Cocos Island meant we could move animals back and forth without any problems. So just in conclusion, next generation sequencing is the wave of the future. The cost of doing it is getting less and less, and the computational technology, which is a major limitation right now, will improve, making it easier and faster for us to do the kinds of analysis that we need to do to sort out what's present there. And this will be used as diagnostics, and it's also going to be used for novel pathogen discovery. But what's most important, I think, is just because we find a pathogen, whether it's known or novel, in a lesion or in an animal, doesn't necessarily mean that it's pathogenic or it's causing disease in that animal, and that a lot of traditional methods and a lot of hard work still needs to be done after the discovery of the pathogen to determine its significance. So thank you for having me tonight, and I'm happy to open up and talk to you about any questions that you might have.